I think many people are aware that she served as the 11th director of the National Science Foundation from '98 to 2004. She's been president of a large number of societies and academies. She has some 40 honorary degrees. In 2007, she received the National Medal of Science from the White House and George Bush. This week, she had received the prestigious Stockholm Water Prize for her contributions to solving water-related public health problems. I'm intentionally not going to talk about the science and all the great things that she's done, because I think that's what she's here to do. She's currently a distinguished professor at the University of Maryland, as well as at the Public School of Health at Johns Hopkins. And I can go on and on and on, but I think rather than you hearing from me, I think it's more important that we hear from Rita and all the great things that she's done historically and continues to do moving forward. So we welcome you, and I think there's no fitting end to this meeting than having Rita talk with the title of Solving Problems with Sequence, which is really what this is all about.
Thank you. Well, thank you very much for all of you who remained to the very last. Indeed, the whole week has been solving problems using sequencing and genomics. What I'd like to do is switch to the field of public health, but I'm going to take a global perspective. I think it's really important to understand that infectious diseases do not carry passports, that we do have intercontinental effects, and one of them is, of course, the plume of dust that comes off the Sahara that goes across the Mediterranean, and from the Gobi Desert that comes to the western part of the United States, carrying a great deal of microorganisms. We're in the process of just starting a project with NOAA to do metagenomic analysis of these microorganisms transported by air. But in any case, what I really want to talk about today is cholera. This is taken from the New York Times about 110 or so years ago, where the specter of cholera was expected to come in from ships to different parts of the world. And as the result of the discussion today, I hope I will disabuse you of the fact that cholera is spreading, but in fact arises de novo. And it provides a really good example of waterborne diseases. So I'll spend a lot of time talking about this bacterium. The organism, Vibrio cholerae, was first isolated by Pacini in about 1880 and subsequently described by Robert Koch. This is a description of a slide from Finkelstein, which shows how it attaches to surfaces and forms both biofilms and attachment structures. And this shows how it attaches to the villi of the intestine. It's a slide from John Clements at Tulane University. Cholera is a worldwide phenomenon, and the darker the color, the more intense the epidemics. And in 1991, there was a massive outbreak in Latin America for the first time in 100 years, which we eventually showed to be associated really with the El Nino phenomenon. But I'll get to that in a little more detail. But the home of cholera has really been considered to be the countries of Malaysia, Bangladesh, and India. And in particular, the Bay of Bengal has been a site that most of the cholera cases have been derived. It's an area of mangrove swamps, and it turns out that the inhabitants of the mangrove swamps are very important. But much of the work that we have done over the last 30 years is in Bangladesh, near Dhaka, in a village area called Matlab, where the villagers have excavated the ponds around which they have built their homes. Just to give you a demonstration of how devastating the disease can be, note that in 2007, there were 150,000 cases. However, missing is Bangladesh, which if you added for that year, the number of cases would be well over 200,000, and the number of deaths would be more like 10,000 or more. But also, you will note that Zimbabwe reported only about 4 cases that year. However, with corruption, with the loss of funding being diverted away from chlorination and filtration, what happened was a devastating result. That is, starting in August 2008, there were about 100,000 cases. As of today, there are well over 110 or 20,000 cases and more like 20,000 deaths just in that one country alone due to the devastation of this disease. I really like the statement of Hippocrates in the 4th century BC, who said, "Whoever wishes to pursue the science of medicine must first investigate the seasons of the year and what occurs in them." And that we have done with cholera. The story of cholera and the waterborne diseases traces to Chesapeake Bay when I first joined the faculty at Georgetown University, and then 6 years later as a professor at the University of Maryland. The work really was a series of studies that were done by a number of students in the Chesapeake Bay at 4 major stations up in the Susquehanna Flats and in the Baltimore Harbor, Kent Island down in the south of the bay, a field station that the Smithsonian runs mid-bay, and then some of the studies done in the lower part of the bay. To make a long story short, some 15 years' work, we found that the bacterium actually has as its host the copepod, and that its abundance is derived strictly from the abundance of the copepod. We were able to show in a whole series of studies, but this is one very good summation of work by Valerie Lewis, a former student. But you can see that the yellow is the temperature, seasonality over a given year in Chesapeake Bay. And then the blue line is salinity, fairly stable. And then the pink line is the chlorophyll. And then these lines are in the different areas of the bay, the incidence of Vibrio cholerae. Now, we don't have cholera in the bay, but the bacterium that causes cholera still is part of the natural bay flora, as it is in all temporal and tropical bays, estuaries, and river systems. The important thing is that the bacterium is part of the natural flora. One of my students, Brad Haley, did a Fulbright in Iceland and was able to isolate Vibrio cholerae from some of the bay areas that receive the geothermal effluent that's heated. So in that warm water, Vibrio cholerae can be isolated, even though there has never been in the history of Iceland any case of cholera. So it's a bacterium that's part of the natural flora. But the spring and fall potential periods of time, should there be some sort of serious breakdown in the public health system of the Maryland, Virginia area, we could have some cases of cholera. We do have outbreaks of perihemolyticus, Vibrio perihemolyticus, and Vibrio vulnificus deaths, 1 or 2 every 3 or 4 years in the bay, and it's part of the Vibrio tracking system of the Vibrios that are in the bay that I'll discuss at the end of my talk. But in any case, we were able, over this 25-year period, to develop a paradigm, a model for the transmission of cholera, showing that it's associated with zooplankton, which become very abundant after the phytoplankton blooms that occur with warmer water, more sunlight. The phytoplankton then serve as the food for the zooplankton, and the cholera are part of the natural flora of the zooplankton. When ingested with untreated water, then human cases occur, and certainly fecal shedding then leads to the person-to-person transmission. One of the other observations we made very early on, back some 25 years ago, which is still kind of contentious, is that between those periods of time when we could not isolate the bacterium, we were able to show that it's present, but it's in a dormant stage. You can detect it with a monoclonal antibody, fluorescently labeled, but our critics said, well, they are probably cross reactions, in any case, they're probably dead. Well, it turns out if you add malodixic acid and yeast extract, you do end up with these organisms being able to uptake radiolabeled substrate and metabolize it, release the radiolabeled CO2. So this is, in fact, a dormant stage. And what we have observed, and many others, there are probably about 1,000 papers in the literature on viable but non-culturable, or the dormant stage, predominantly of gram-negative bacteria that don't form spores. And on a heterotrophic plate count at low temperatures, like the winter months, after 3 or 4 days, you can no longer grow anything from the culture that's been exposed to low temperatures. Perhaps a little bit longer in a broth culture, but the direct viable count shows that there is some decline, but still very large numbers of the cells are viable. And the acridine orange count shows that all the cells are there, even though not all the cells respond to the nalidixic acid. Again, the critics said, well, they don't really cause disease. So we were able to show by rabbit loop inoculation that indeed you can recover the cells 4 or 5 days after inoculating into the rabbit loop the Vibrio Cholerae. And then the comment was made, well, that's animals, it's probably not humans. So we were able to do some human volunteer studies in the Center for Vaccine Development, Mike Levine group at the University of Maryland Medical School. And we were able to show that with about 10 volunteers, that after about 5 days of having ingested a culture that was viable but non-culturable, not able to be cultured, but the cells were still there. And also, it was a strain from which the cholera toxin had been excised. We didn't really want to create a problem in the Baltimore area. But after about 5 days, we were able to culture from the stool of the volunteers Vibrio cholerae. And at 5 days, we're, of course, they are given antibiotics and made sure that when they are released at the end of a 2-week period, they're not turning out to be the cholera Johns and the cholera Marys of Baltimore. Now, most of the work has been done in Bangladesh, in Madlab, in the villages of Bakarganj, right on the coast of Bangladesh, because, as I say, that's a mangrove swamp area, and those who study plankton, phytoplankton, and zooplankton tell us that mangroves are where copepods are dominant. They'll probably be 90% of the zooplankton population. Village houses around those ponds that you saw earlier in the rotating earth, the houses are built around the ponds, and these ponds serve as a source of water, drinking water, washing sites, as well as, unfortunately, latrines. The bacteria, of course, don't behave themselves and stay in the latrine corner, but migrate throughout the pond. The victims are predominantly children under the age of 5. Diarrheal diseases, waterborne diseases are the major killer, second to only acute respiratory infections. And you can see a direct correlation. The fewer the people that have access to safe water, the larger number of deaths of children under the age of 5. The U.S. having almost every one of us having access to safe drinking water, very low mortality rate to diarrheal disease. A cholera victim, still this cholera cod is used. We measure the fluid lost through vomiting and diarrhea, very crude, but it's a measure of what needs to be replenished. So this is the background for the genomic work that I will be discussing in a moment. The bacteria are associated with copepods, and shellfish feed on copepods, and then the end result with poor sanitation is a person-to-person transmission, but really the source is from the environment, consistently from the environment. The environmental signatures that are related to cholera epidemics are part of the story and part of the ultimate genomic tracking that we are now in the process of doing. Clearly, the annual cycles of sea surface temperature warming that occurs in the spring and that in the Bangladesh region occurs in the fall, this coupled with the changes in the sea surface height, which are the tidal patterns that drive the water from the Bay of Bengal up into the river system, and it is a brackish water system, so that the plankton that are present in the Bay of Bengal then contribute, then you can see the patterns of the plankton populations, the abundant spring and fall. So these global patterns have provided us with the ability to understand these waterborne diseases, particularly those that are transmitted by plankton populations. It occurred to us back in the mid-'80s that we could probably monitor cholera by measuring chlorophyll using sensors and satellites. And in fact, this was quite successful. This is one of the very early studies in 1994, where we were able to show that as the temperature, I hope you can see it, the blue line, as the sea surface temperature increases, just as we had predicted, there would be about a 6 or 8 week lag from the peak in phytoplankton to the peak in zooplankton, and then the distribution of Vibrios into the water column. And we have subsequently done a series of studies showing that this relationship really is very, very significant. The environmental signatures that are related to cholera epidemics, you can see the direct relationship for Calcutta, India, and Madla, Bangladesh. In fact, we are able to show a very significant positive relationship between cholera, chlorophyll, and rainfall. In fact, in Calcutta, when you have a milligram per cubic meter increase in chlorophyll in the water system, you have a 33% increase in the number of cholera cases. And when you have a millimeter per day increase in rainfall, you'll have about 7% increase in the number of cholera cases in that city. Similarly, in Madla, Bangladesh, when you have a milligram per cubic meter increase in chlorophyll, again, about a 31 or 32% increase. And we're now looking at earlier predictions of the capacity to predict cholera. And summing the data, these cholera relationships and the prediction of cholera based over a period of time from 1998 to 2005, we can see that our predictions have been very accurate from the model, both for Calcutta, Madlab, Africa, Ghana, and for Peru. The data for Peru are less, they're more scattered because the data gathering is not as complete as it is for the other countries. More recently, and this now gets into some of the genomics, the outbreaks in Senegal in Africa have been very, very severe. It turns out that the number of cases are in the 20,000 in 2005 and continuing. But what's very severe about it is that the deaths are more like 20% instead of 2% of the cases. And it turns out in the studies we've done that there is a duplication in the strain that's rampant in Senegal of the cholera toxin gene. But also these factors, these environmental factors, are working in Senegal as in Bangladesh and India. That is, there has been a sea surface warming off the coast of Senegal and a sea surface height anomaly as well. And that accounts, we think, for this relationship. Now, in the year 2000, John Heidelberg, a postdoc, a former student and a postdoc at Tiger, and Jonathan Eisen, who's here in the audience, along with Claire Fraser and the team at Tiger, and collaboratively with my lab at Maryland and John Mekelanos, were able to sequence Vibrio cholerae and demonstrate definitively that it has 2 chromosomes. Now we know that all Vibrios have 2 chromosomes, but that became our reference strain. More recently, through the collaboration, the wonderful collaboration with JGI, with Tom Breton, Chris Detter at Lannel, and the folks here at the JGI, we were able to sequence 35 Vibrios, and we were able to sequence more than 20, about 23 Vibrio cholerae. These are strains that are from 1910 from Saudi Arabia, isolated in an outbreak then, from 1975 from an outbreak in Bangladesh, clinical strains, a water strain from the US Gulf Coast that was isolated in 1980, a sewage strain from Brazil in 1980, a whole series of historical strains, widely geographically distributed from water systems, from patients, from food that had led to cholera outbreaks, and even from Chesapeake Bay plankton. This allowed us then to compare the non-Vibrio cholerae with against we're calling it the reference strain, the 2000 strain that the Heidelberg, Eisen, et al. Strain, and compare the other Vibrio cholerae. What was very interesting is that no one strain of Vibrio cholerae whether historical or recent, was identical to every other strain. They all have some variants. And furthermore, they also lack, you can see that the genes that are not present in the Vibrio, that's a photobacterium, and then there's Vibrio farnesiae, Vibrio parahemolyticus, etc. What's also fascinating is that we were able to show quite clearly that the vulnificus and the parahemolyticus probably, we're in the process now of sorting this out, probably deserve a separate generic status, but we are just in the position now of doing these analyses. Furthermore, we had isolated a couple of Vibrios from the Chesapeake Bay that we thought were cholera, but these turn out to be new species. And the Vibrio cholerae strains, L-Tor, classical, and so forth, clustered together. But when you analyze all 23 Vibrio cholerae and the Vibrio mimicus, we find some very interesting metabolic differences. And this, I think, will lead us to being able to do a metabolic biogeography of the Vibrio cholerae isolates from around the world. In other words, the locale from which the strains are isolated have their own specific metabolic characteristics from the laterally transferred genes that allow this kind of incorporation of metabolic capacity. And that, again, to emphasize the diversity that occurs within what is called a single species of bacteria. We've been able to track exactly the changes of the cholera isolates that we have sequenced, looking at the genomic islands that code in yellow, that code for the O antigens of the O1 serotype, and the major pathogenicity islands as well. That's shaded in a kind of a light pink. I guess it shows up as a darker red. And then the strain-specific genomic islands, and we can follow the changes that have occurred in every one of the isolates of Vibrio cholerae from around the world in different locales. Essentially, an evolutionary path of this one single species of cholera. What has been particularly interesting is that the organism is tracked, monitored by the O1 serotype. This has been considered to be the very most important characteristic of the cholera Vibrio. Well, when you look at the genes that code for the oligosaccharide of the O1 antigen and the genes that code for the antigen itself, we find that there are a variety of variations that lead to the different subtypes, but the most fascinating part of it is that the O1 antigen, the O1 genes are laterally transferred. In other words, there are several different backbones, and the O1 cassette moves around the Vibrio population. And this explains, I think, the outbreaks that occur episodically in various parts of the world. In addition, we have found that the core genes of the Vibrio seem to be located globally, but the metabolic changes are quite geographically specific. In fact, we have proposed that just as in with viruses, that there is a genomic drift and a genomic shift that occurs amongst the Vibrio cholerae, which I think would explain as well the fact that there's been no real success in obtaining a vaccine for cholera. It is constantly changing so that a vaccine at best will be 60% effective in a given locale. In fact, most of us, years ago when we were told to get the vaccine for cholera when we traveled to cholera countries, I remember Mike Levine telling me, don't bother, I'll just fill out your passport form that you've been vaccinated because it isn't any good anyway. More recently, there are some oral vaccines that have been proposed, and one has been clinically in clinical trial. But still, I think the problem lies more in the genomics of the organism and understanding better the Vibrio cholerae and producing perhaps DNA-type vaccines rather than a surface vaccine. Finally, let me just point out that we are continuing these prediction capacities, and we have been able to utilize a model for the Chesapeake Bay that was developed really to monitor harmful algal blooms. But using the model and modifying it for Vibrio cholerae, we were able to actually get a probability map of where in the bay you would pick up the organism, most likely at given times of the year, when the conditions, for example, are appropriate. And here are shown over a period of time from 1991 through January to 2005 for 3 different locations in the Chesapeake Bay and the times of the year when you take into account the environmental parameters, you're most likely, the highest probability of having cholera present, Vibrio cholerae present in that water system. So that allows us then with these anomalies of V cholerae probability, that is, when it is most likely to be present above the line and less likely to be present based on these environmental parameters. And so we then will have an early warning system for Vibrios in general, not just cholera, but Vibrio perilyticus, vulnificus in the bay, and the model is extrapolatable to other parts of the world. So I conclude by saying that, you know, when you tug on anything in nature, it's hitched to the rest of the universe. The words of John Muir, born in 1838, died in 1914, and the founder of the Sierra Club. And I'm very proud of the collaborators from the International Center for Diarrheal Diseases Research in Bangladesh, physicians, microbiologists, sociologists, many, many students from the University of Maryland, colleagues and visiting scientists, and collaborators with NASA Ames, the late Louisa Beck and Byron Wood, 2 wonderful people, and Brad Lobitz, who continues collaboration with us at NASA Ames. The Esri folks have been very helpful, and colleagues from other countries. In fact, my lab is international. It's the United Nations at the University of Maryland. And I would like to thank you all for the opportunity to talk a bit about how you solve at least this problem with genomics. Thank you.